Hi, my name is Claire Green, and today I'm going to be presenting on the paper an analysis of polyethylene terephthalate degradation kinetics of evolved ISPTA's variants using a surface crowding models. This was a paper published in the Journal of Biological Chemistry, and it had numerous authors, the first author being Zhang and Johnson. So a little bit of background information about kind of what this paper is coming from and why it was important when it was published is that plastic waste is an ever-growing problem on our planet. In 2023 alone, the world produced 413 million metric tons of plastic, according to Statista in 2024. What this means is that every year we're producing millions of tons of plastic, and there's really nowhere for that plastic to go. It's ending up in landfills, it's ending up in the ocean, and it's causing problems because it's accumulating at such an incredibly quick rate. Plastic degrading bacteria were actually discovered back in 2001, but nobody really saw plastic waste as a huge problem back in 2001, and so publications didn't emerge until 2016. So through this presentation, we're going to kind of find out why and how these degradation kinetics occur and see what processes happen. So some more background info. Polyethylene terephthalate, or PET, has been around since the 1940s, but we didn't really see major discussion of plastic degrading until 2016. PET is produced by the polymerization of ethylene glycol and terephthalic acid, and research in 2016 by Yoshida et al. Found a bacteria called Ideonellus gaensis can degrade certain types of plastics, including PET. The enzyme that they found connected to Ideonellus gaensis releases soluble PET degradation products, and there's a list here on the screen of them. They include ethylene glycol, TPA, mono-2-hydroxyethyl terephthalate, or MET, bis-2-hydroxyethyl terephthalate, or BHET, and potentially oligomers as well. So that's a quote from the paper that I'm discussing today, but that original paper in Yoshida et al. In 2016 is what kind of discovered those soluble products. And here's a figure from that Yoshida et al. Paper in 2016 that shows those degradation pathways. It shows kind of what breaks down into what and how we end up with these various products. And I found it very helpful to just kind of see the visual of how one kind of bigger thing becomes a lot of smaller things. Now, of course, becoming smaller and breaking down is what we really need to see in these natural systems where we just see plastic building up as it is and staying in that PET form. Some other terms that you kind of see throughout the paper and in the title itself are a surface crowding model and kinetic analysis. So a surface crowding model examines how enzymes work and interact in crowded and complex environments, and kinetic analysis is a method to study effects of mutations on catalytic activity, to see kind of how things interact with each other and break down, and how when we mutate specific genes or portions of enzymes, how that changes that catalytic activity. Oftentimes, kinetic analysis uses the or Michaelis-Menten equation, but in cases like this one where we have surface crowding, where we have kind of complex interactions going on, you have to oftentimes use an adapted Michaelis-Menten equation or various multiple equations in order to get more of an accurate picture of what's happening. The question that the paper kind of looks at is why can ISPTAs successfully degrade PET, and how do mutations affect its productivity? The authors did hypothesize that ISPTAs displays enzyme concentration-dependent inhibition effects because of surface crowding. So they said that the amount of enzyme matters because when you have too much enzyme and not enough surface area for that enzyme to interact on, adding more enzyme is not really going to change your outcome or be beneficial whatsoever. So having the proper amount of enzyme for the proper amount of surface that you have creates the most efficient circumstance. They didn't really make a hypothesis about which mutations exactly they thought would work or how they would work. They just said, here's a few that we're going to try, and then we'll get into some of those results later on. They had a variety of methods and quite a long process that they went through in this paper, but I'll try and walk you through it. So they started by cloning and constructing plasmids. So when you're cloning and constructing plasmids, you're essentially taking a specific area or a sequence of DNA and you are editing it to be what you want it to be. So in this case, they were mutating specific areas of the sequence to have various effects that they later tested to see exactly how they affected the productivity and effectiveness of this enzyme. But when you've got that sequence in there, then you're just going to clone it, or you're going to create a bunch of different copies of your mutated sequence. And then they inserted a disulfide bridge, and disulfide bridges are where you have two sulfides from cysteine that are connecting, and they're adding kind of stability to a structure. And the stability can be beneficial, or it can make things more rigid. So we're going to see a little bit kind of the effects on flexibility later on. Then they took these modified constructs or plasmids and they transformed them into E. coli. So E. coli is often used in these studies where we're modifying something, or we're looking at enzymes, or we're looking at kinetics, because E. coli A grows fast and B is pretty easy to keep alive. And so we like it because it's pretty hardy, and so you can pretty reliably say that your E. coli is going to survive, and it's going to allow you to visualize specific areas of DNA or of these plasmids. And then they took a look at thermal stabilities of purified enzymes using differential scanning fluorometry. And when we're looking at fluorometry, what we're looking at is what is fluorescing under various circumstances. And so in thermal stabilities, what we're testing is various temperatures. They're taking a look at one temperature and seeing what's fluorescing at that temperature. Then they're taking a look at a higher temperature and they're seeing what's fluorescing at that temperature. And when you see the differences in what is and isn't fluorescing at various temperatures, you're seeing when you have a stable product and when you have a product that's breaking down. Then they took a look at these enzyme kinetic reactions, and they conducted these and used supernits. And so they measured essentially what was going on by the amount of materials that were used in these breakdowns and in these pathways. And then they looked at tagging percentages, again using fluorescence, so they were trying to see what percentage of things were tagged. And then they also looked at ISPTase absorbance on PET, and they measured that again using fluorescence. So they put this ISPTase on polyethylene terephthalate, and then they used this fluorescence to see how much of it was being absorbed and how much of it was just sitting there not being absorbed. So now onto kind of those initial results. They found that random and semi-rotational directed evolution, or various modifications that they made to the enzyme of ISPTase, identified 3 different mutations that enhance purification yields with potential benefits to activity. And we're going to walk through each of those 3 mutations that they found. This is just a good overall figure from the paper that shows you kind of what was going on. That previous slide had also a quote from the paper that was telling you about those 3 pathways and what went down. But this shows you how the temperature was affected, and it also visually shows you those 3 different mutations, kind of where they were. They're in different colors here, which is really helpful. And then we'll see what exactly those mutations did. So taking a look at that first mutation, or the 116P mutation, it revealed a 30% improvement in maximum product accumulation rate. So what that means is more product was accumulating, more reaction was happening, and this was because it decreased susceptibility to crowding. So in the case of this mutation, they said, well, this doesn't actually make the reaction itself more efficient. It doesn't actually really change what's going on kinetically, but it reduces the susceptibility to crowding, so it means that the enzyme can work in more environments. And because it can work in more environments, we see that more product is going to accumulate overall. And that 30% is kind of a rather large number in the context of this paper. Then they took a look at the TSP S238, and they found that that one had a cumulative 1.5 degrees Celsius increase in thermostability, and that this resulted in a small reduction in maximum productivity. So there's kind of a pro and a con there. They saw that it could work in higher temperatures, it was more thermostable, but they also found that it was slightly less productive compared to the not modified version. And then finally, S290 was introduced, and this displayed another 1.5 degrees Celsius increase in thermostability, with slight reductions in maximum productivity, and this mutation was also found to increase elution yields. And so because it increased elution yields, it was something that was overall beneficial, and researchers were like, this was a good overall change. And then to kind of summarize a little bit, they found that the simulations demonstrated that crowding led to reduced overall enzyme flexibility and active site residue flexibility in free enzymes, while hindering the formation of productive conformations in substrate-bound enzymes. So what does this mean? This means that flexibility, the ability of these enzymes to fit in these different environments, went down, and that also meant that the ability to form these productive conformations, these good conformations in these bound enzyme types, went down. So we found that crowding overall was pretty bad for the enzymes. They found overall that evolved enzymes were more efficient than the original enzyme, and just kind of throwing back to that very first mutation that had that 30% improvement in maximum product accumulation rate, that's an example of how that occurred and how that evolved enzyme was better. This figure takes a look at that flexibility. It shows you how flexibility changed in these various conditions, and it also offers a good visual. You can kind of see what's going on up here. And just this, again, was how flexibility decreased when there was an increase in crowding. And so crowding was harming the flexibility and the ability to make those good conformations. So a new catalytic model was also developed for analyzing this ISPTase activity. So when we got all of those numbers from those mutations, it was because they had this new catalytic model. And I talked a little bit about how the Michaelis Menten doesn't account well for many types of enzymes because it doesn't account for these complex environments that they're working in. And so these researchers set out to kind of fix that. They were like, we don't have an equation or a model that we can use that accurately shows you what's going on, and so we're going to make one. They did a bunch of math, they did a bunch of deriving, and this is kind of what they came up with. This is a screenshot from the paper, and it just shows you the functions that they used, and the paper includes exactly how they got there as well. So onto that critique. So it was unclear why the authors chose the specific mutations that they did. Clarification of why they chose the specific mutations that they did would offer a lot more analysis into how we got to where we are. How they decided that these were going to be the most important or the most promising or the most beneficial would be interesting, because then that can be applied to choosing enzymes in the future for other papers and other projects. And a clarification of this and a more clear way to reference the mutations would be helpful. The mutations are kind of referenced throughout the paper with these long numerical forms, and just giving them kind of a key, saying maybe mutation A, B, and C would be helpful because you're not searching through to find those various numbers. And then more analysis into why the mutations reacted the way they did would also have been beneficial. They told us, these mutations were beneficial, these mutations were more effective, these mutations were more thermostable, etc., but they didn't say this is why we think this is more thermostable. And understanding why those processes occurred, why we saw the changes that we did, would again be helpful in designing future modifications, designing future transformations, seeing if these are modifications that we should try and go put in other enzymes, or if these are modifications that we should try and do all at the same time. Like, should we modify all of these things at the same time to see even greater benefits? The future implications of the study are that the study sets up the idea of using these mutated sequences in both engineering and biotechnology, and we have seen a lot of research recently that looks at various bacteria species and enzymes in bacteria that break down various types of plastic. So not always PET, sometimes we're looking at things that break down polyethylene, for example, but there's also numerous other polymers that are associated with plastics, and numerous other bacteria that are associated with degrading those polymers. And so being able to apply this idea of mutating sequences that we find in nature could be applied to all of these other bacteria and all of these other types of plastics. And so hopefully the study will be recreated with other enzymes and other plastic degrading organisms, and hopefully those other studies will see similar results, where when we do some of this engineering, when we do some of this biotechnology, when we do some of this mutation, we see that that makes a product that we can go out and use and that can be more efficient at kind of solving our plastic waste problem. Thank you for listening to this presentation. It was created using a template by SlidesGo, including icons by FlatIcon and infographics and images by FreePic. Then here's a references page. Most of these references were quoted directly somewhere in the presentation. One that was used for background information was Britannica, that gave some background information about the history of plastics like polyethylene terephthalate, and the others are quoted directly where they're used in the presentation. Thank you.